Well, hello again, everybody. Welcome back. Today we are going to look at one of the longer chapters that we covered this semester, I think. That's my impression, because there's a lot of information in this chapter. It's looking at basically what a typical cell looks like. We will talk a little bit about bacterial cells, but we're not going to spend a lot of time focused on them. We're going to spend more time focused on eukaryotic cells. And we could get into a whole lot of detail, but I want to try and keep this presentation focused on what we need to talk about, the actual organelles in the eukaryotic cell. And hopefully we can do that in a reasonable amount of time. Now, I have broken it up into a number of very small chunks along the way with periods of break with a concept check question in there, as I've been doing with the previous chapters. And you are welcome to stop at any point along the way when we get to one of those breaks. It's a convenient place to step away, take a bathroom break, grab a drink or a snack or something like that, or just simply walk away for a while and come back and take another look at it. As I've said previously, if you put me on pause, if you turn me off, it really doesn't matter. I won't know, but I will still be here when you come back again. And so we will progress through this presentation in much the same fashion as we've done previously. And hopefully we can keep this to a respectable amount of time, roughly around 2 hours, maybe 2.5 hours. I really don't want these presentations to go much longer than that, because I do realize time is one of those things that we do have to be careful with. So that being said, let's get started. So when we start thinking about cells, when we start thinking about organisms, we have to bear in mind that the smallest unit of life is a cell. So all of those properties of life that we've talked about previously, they are all encapsulated in a single cell as much as they are encapsulated in a living organism. And in fact, some organisms are single cells. So not surprisingly, we would really expect to find all of the properties of an organism encapsulated or found within a single cell, because some cells are single organisms. And there's an idea in biology, it's actually a theory called the cell theory of life, which includes that concept. The cell is the smallest unit of life, and it adds an extra part to that, that all current cells come from pre-existing cells, which has the implication that cells are able to copy themselves. In other words, they're able to divide and make new cells. And in a later chapter, we're actually going to address the processes of cell division. We're not going to talk about those so much in this chapter, but we will talk about the ability of cells to replicate themselves in future chapters, or a future chapter, because that becomes an important part of the continuity of life, and it has implications for a number of other things that we know about living organisms as well. But more of that in another chapter. That's a few weeks down the road yet, before we get to that chapter. But we start out by thinking about antibiotics and the fact that antibiotics have the ability to inhibit cell activity. And they can either focus on the biochemistry of the cell, or they can focus on dealing with some of the structures that are found in cells, most notably bacterial cells. The term antibiotic means anti-life, but it relates particularly to bacteria. And that's one of the things that we always need to remember about antibiotics. They don't have any effect on viruses, because viruses are not living organisms. So something that's anti-life is not going to affect something that's not alive. So when you have a cold, the flu, COVID, or any other virus, an antibiotic is simply not going to help you. Antibiotics only work against living cells, and primarily they only work against bacteria. That's what they're designed for. The problem is that bacteria also have the ability to gain immunity or resistance through mutation to antibiotics, largely because of the way that we use them. but it is also a function of the cells themselves. We know that organisms are able to evolve, and it is the environment and the challenges of the environment that trigger that evolution, by virtue of the fact that any organism that has some sort of advantage over the surrounding organisms, or in this case, surrounding cells, is more likely to reproduce, thrive, produce lots of offspring, and those offspring will be just like itself or more like itself. That is, having the same advantage. That's the process of evolution. And in this case, adding antibiotics to the bacterial environment simply provides them with an opportunity to evolve, to become resistant to it. And that's what we've experienced over time. That bacteria have actually developed resistance. But there's a greater problem than that. The fact that bacteria can share the genetic solutions to antibiotics with other bacteria. And so even if some bacteria have never seen an antibiotic, they can have that resistance to that antibiotic before they even see it, because they've picked it up from another bacterium that already has antibiotic resistance. They don't need to develop it for themselves. They can simply capture it from someone who already has it. And so this poses a real problem for us in terms of trying to deal with bacteria. But bacteria themselves represent one type of cell. They are not the types of cells that our bodies are made up of, but they do represent a type of cell that you will find in your body, because whether we like it or not, there are many hundreds of species of bacteria that call our body home. And they're located in all sorts of places, in the mouth, in the stomach, in the small intestine, the large intestine, the colon, throughout the body. And it turns out that we actually need them. So one of the things that we also need to be careful of is that when we do use antibiotics, because some antibiotics are referred to as broad spectrum, meaning they target everything. They're not very specific in the types of bacteria they target. They basically target everything. Those sorts of antibiotics, as much as they may be beneficial in curing a bacterial disease, they can do a number on the beneficial bacteria that are in our body. And that can also have consequences. And sometimes it takes a while for our bacteria to return to their normal populations again. And that can actually leave our bodies exposed to all sorts of issues, because those bacteria not only live in our body, they're beneficial to us. And one of the benefits is they simply protect us from the more unfriendly bacteria that would love to make a home in our body, but would in turn make us very, very sick. But because the friendly bacteria in our body outcompete them, for now at least, and our immune system can keep them in check, and we can use antibiotics to get rid of the bad guys, our beneficial bacteria really do help protect us. And there is an increasingly large body of work demonstrating just how important the bacteria that live in our gut are to the health of our body. We are now recognizing that a large portion of our immune system is associated with our gut. And it talks, and it talks backwards and forwards. Our gut immune system talks backwards and forwards with the bacteria, particularly the friendly bacteria that live in our gut. And those bacteria can also communicate with our brain. And there is communication between our brain, our gut, and the bacteria. And it is a very, very important communication pathway. We don't have a lot of time in this presentation to talk at length about this, but that might be something that you want to investigate for yourself. This is becoming a very, very interesting area of research. And if you've ever heard the term prebiotic and probiotic, you're starting to hear the terms that address this relationship between our body and the gut bacteria that inhabit our body. And prebiotics are simply chemicals, food molecules that we can consume that are beneficial to the, prebiotic of the bacteria, Probiotics are the chemicals that we can include in our diet that promote the growth of the bacteria. And they work together along with other components in our body to maintain the health of our body. And it may even have something to do with some of the diseases in terms of obesity, our moods and things like that. The information that is being gathered from research about our gut bacteria is pointing to the importance of these bacteria in some of those areas of investigation. Again, lots of interesting things. We really don't have time to talk at length about them. So that might be something that if you're really interested, you want to do some research on for yourself. But let's start talking about the different types of cells and then start focusing in on eukaryotic cells. So we've talked about the fact that, or I've mentioned that there are two different types of cells. So we refer to those as prokaryotic cells and eukaryotic cells. Prokaryotic cells are typically the cells that are associated with two of the three domains of life, domain bacteria and domain archaea. And these are the two domains of life that are made up of, or that contain organisms that are prokaryotic in nature. And the term itself actually gives us some clues as to what the meaning is. So the term karyon is from the Greek language, and it refers to the nucleus. And another word that we could use for pro is pre. And so when we say prokaryote, we're referring to something that is pre the nucleus. In other words, these are the cells that predate the origin of the nucleus. These are cells that lack a nucleus. The other type of cell is called a eukaryotic cell. So again, we have karyon involved, and it's talking about the nucleus. The prefix "eu" has a number of definitions, and I've talked to one of my colleagues that teaches Latin at Valencia, and he said there are a number of different ways that you can interpret this. And one of the ways you can interpret it is to say true or real. So when you see the prefix "eu" ahead of the term karyon or karyotic, which is derived from the term karyon, we're talking about a cell that has a true nucleus. And so one of the defining features of eukaryotic cells, the cells that are found in plants, animals, and fungi, the three big eukaryotic kingdoms of domain eukarya, you're talking about cells where there is a visible nucleus in the light microscope. And these definitions are based on what you can see in the light microscope, which was, of course, the original way of being able to view the cells once we had microscopes available. And so with prokaryotic cells, when you take a look at those in a typical light microscope, you struggle to see more than just the barest outline of the cell shape. That's it. They're really, really tiny. Whereas eukaryotic cells tend to be larger. And when you look at them under the light microscope, you can actually see the nucleus. It is the feature that is most prominent in all eukaryotic cells once you get them under the light microscope. That being said, that doesn't mean that some prokaryotic cells don't get really big. The largest bacterial cell is almost 0.5 millimeter in length, which makes it almost, it's right on the very, very limit of what you can see with your naked eye without assistance from a lens like a magnifying glass. But the average prokaryotic cell is never that big. That would be where we would start thinking about eukaryotic cells. And some eukaryotic cells do get large enough that you can actually see them with the naked eye. Barely, but you can see them. But again, most eukaryotic cells, and certainly the cells that are present in our body, are better viewed with the light microscope rather than with the naked eye. So size is a function of the two different types of cells. The reason why size comes into play and why there is such a difference in the typical size of plant cells and animal cells and eukaryotic cells in general versus prokaryotic cells is because of what we see inside the cells. So when you finally get a chance to look inside a bacterial cell using an electron microscope that can magnify tens of thousands and certainly several hundreds of thousands of times with a really good one, what you recognize is that the inside of a bacterial cell, a typical prokaryotic cell, is not subdivided into smaller compartments, whereas all eukaryotic cells are. And that makes a difference because once you start dividing the cell up into smaller compartments, you create the opportunity to support lots and lots of chemistry because the amount of membrane that you have available is correlated with the amount of chemistry that you can support. So if the only membrane that you have is the cell membrane, the membrane that surrounds the cell, if that's the only membrane that you've got a a limited amount of membrane. And as the cell gets bigger, it turns out that the volume of the cell increases faster than the surface area of the membrane. There is a mathematical reason for that. We won't get into it. But as you increase the size of the cell, the surface area of the membrane increases, but the volume increases even faster, which reduces what is known as the surface area to volume ratio. So when a cell gets bigger, you decrease the surface area to volume ratio, and that has a direct effect on the cell's ability to carry out different types of chemistry and support different types of chemistry. But if you then take a cell and divide it up into smaller compartments, you're actually adding more membrane, which starts to increase the surface area to volume ratio again. Actually, I think I'm talking about it in reverse. When the cell gets bigger, the surface area to volume ratio gets... No, I'm talking about right. It depends on how you want to view it. But suffice to say, there is more surface area, membrane surface area, if you divide the interior of a big cell up into smaller compartments using membranes. And that's what we're going to see in eukaryotic cells. The division of the inside of the cell into smaller units by using membranes. And that increases the amount of membrane that you've got available and the surface area of the membrane that you've got available, which allows you to support more chemistry and to do so in a more effective and a more efficient fashion, which is the reason why eukaryotic cells can get bigger, because they can support the same or increasing amounts of chemistry because they've got more membrane to do so as the volume gets bigger. They're not dependent on just the cell membrane anymore. So that's the reason why there is a size difference. There's also a complexity difference as well. Bacterial cells are quite complex, as we can't argue that. Any cell is quite complex. But eukaryotic cells are even more complex because, again, of the presence of these smaller compartments, which we refer to as organelles or little organs inside the cell. And these compartments or organelles allow the cell to then create specialized units for doing parts of the chemistry. And that means that they, rather than being generalists, they become specialists, and they can then focus on doing one thing and doing it exceptionally well, in much the same way as the organs in our own body became specialist organs rather than generalist organs. And so each organ and organ system in our body focuses on doing one thing and doing it exceptionally well to the benefit of us as an organism. And the organelles inside eukaryotic cells essentially do the same thing. That's why we use the term organelle. Remember, it translates as little organ. So basically what we're implying is that an organelle to a cell is the same as an organ or an organ system to a multicellular organism like ourselves, where we have special structures that carry out specialized functions. So that's something that we need to bear in mind as we talk at more length about eukaryotic cells particularly. So I've introduced you to the cell theory of life. We don't really need to talk about this anymore, but here it is stated again, just in case you missed it. So the cell theory of life says that cells are the smallest units of life, the smallest unit that contains all the properties of a living organism, and all existing cells come from pre-existing cells, the implication of cell division. We've talked about the two different types of cells, prokaryotic cells as bacteria and archaea, and eukaryotic cells as everybody else. So plants, fungi, and animals, and then this rather diverse group of organisms that are typically unicellular, like bacteria and archaea, but can be multicellular as well, referred to as protists. And there's a broad diversity of protists. We used to actually have a category for them, a kingdom, like we have a kingdom for plants, a kingdom for fungi, and a kingdom for animals. But more recently, over the last 20 or 30 years, we've recognized that these guys are just so diverse that we really needed to split that up into multiple smaller categories. And people are still trying to figure out, do we want to create many, many kingdoms, or where do we want to classify them? So most people sort of kick the can down the road a little bit and just still continue to call them protists, recognizing that that's a general term. We all know what it means. We have a general understanding of what it means out of convenience, but people are still struggling with what to do with these guys in terms of how to categorize them all. And so these are the eukaryotic cells, and they belong to the domain eukarya. Let me just put this in, just say domain. So remember, we do have the three domains. So we have domain bacteria, and we have domain archaea. And the reason why bacteria and archaea are given their own domains is because there is such a broad diversity of bacteria and such a broad diversity of archaea that they rival in terms of sheer numbers of cells and in diversity the eukaryotic domain. So they really do warrant having their own categories, their own domains. So these are the three domains that we identify, and we've talked about those before in a previous chapter, so this should be nothing new. So here are some diagrams that illustrate the differences between the cells. This is a compare and contrast, and this is something that you ought to be able to do. So make sure that you pay attention to these tables here in the slides, and also in the textbook as you're reading the textbook. Make sure that you pay attention to these, because a compare and contrast question is the sort of question that I might very well ask you in the exam that covers this chapter, which of course is exam number 2. So something that you need to be aware of. One of the things that we do recognize is that there are some features that are common to both types of cells, prokaryotic and eukaryotic. Yeah, there are some differences, most notably the presence of the nucleus. There are others that we can talk about as well, but there are some common features that people recognize. And so we do typically think of 4 common features that are universal to all cells, whether we think in terms of prokaryotic cells or eukaryotic cells. So I'm just going to put a note here and just say these are, that's not very neat. Let me take that out. These are universal to all cells, both prokaryotic and eukaryotic. Now, the note here kind of implies that, but I want to state this specifically so that there can be no confusion. These 4 features are considered to be universal to all cells. So what are they? The presence of a plasma membrane or cell membrane. So let's put in here, AKA cell membrane. The 2 terms are interchangeable, so you can call it a cell membrane or you can call it the plasma membrane. Every cell has got to have one because this is the membrane that demarcates or separates the inside of the cell from the outside of the cell. So every cell has one. Inside is a very thick fluid-like material that has proteins, sugars, fats, all sorts of things mixed into it. And it creates a fluid-like material that we refer to as the cytosol or cytoplasm. So again, we'll put in an AKA. And this is the medium inside the cell in which all the other features that we find inside the cell are actually embedded. So in bacteria, there's not going to be too much embedded into the cytoplasm. In eukaryotes, we're going to see that there's a whole lot of stuff embedded in the cytoplasm. But again, all cells have cytoplasm. And if you want to try and think about what this material is like, the best description I can give you is that it's kind of like that intermediate stage in the making of jello gelatin, where it's no longer liquid. As the water, you know, when you make gelatin, you dissolve the crystals in hot water and then you allow it to cool. And as it cools, it goes from a liquid into a more rigid jelly type material. In the middle of that process, as it's cooling down, the jello is no longer liquid, but it's not solid yet either. It's not into its final gelatin form. And it's that midway point that's the best analogy that I can give you for cytoplasm or cytosol. It's hard to describe it in any other way. I don't know how else to describe its nature other than to use that as the analogy, because that is the best analogy that I can think of to explain what this material is like. It's really weird stuff. Then we do know that DNA is the genetic material, the molecule of inheritance of all living organisms, whether they be prokaryote or eukaryote. So you're going to find DNA as the molecule in which genetic information is encoded, and that's universal across all cells. And there's, as we'll see in some of the later chapters, particularly chapters 11 and 12, when we talk about DNA technology, that has really changed the way we view DNA because of its universal nature. But more to come of that towards the end of the semester. And then because all cells need to have polypeptides and proteins, they all have ribosomes. But we do know that the ribosomes in prokaryotes are very different from the ribosomes in eukaryotes. We can differentiate between them. And there are some interesting observations that we have made about some organelles in eukaryotes, and we'll talk about that a little bit as we get a bit deeper into this presentation, that have suggested some interesting origins for a couple of organelles that we find in eukaryotes. So I'm teasing you there right now, but we will talk about that a little bit later in this presentation. But suffice to say, ribosomes are universal. We find them in prokaryotes, we find them in eukaryotes. There are some differences between prokaryote ribosomes and eukaryote ribosomes, but they do the same thing. They both assemble polypeptides and proteins. So when we think in terms of the evolutionary history of cells, prokaryotes have been around since the origin of life. Our oldest fossils that have been confidently dated and absolutely dated are about 3.48 billion years old. And these are somewhat complex prokaryotic cells because they carry out photosynthesis. They belong to a group of bacteria called cyanobacteria. And they're already carrying out this complex process called photosynthesis, and it's photosynthesis as we see it in plants. It's identical to what we see in plants today. And so that tells us that prokaryotes have been around at least that long, perhaps even longer, because prokaryotes and archaea, or bacteria and archaea, must represent what the very, very first cells actually look like. They must have been prokaryotes, and they probably appeared first somewhere around 4 billion years ago, maybe a little bit further back in time than that. We're not entirely sure exactly when the first cells appeared, but we do know that at 3.5 billion years ago, 3.48 billion years to be exact, if you want to be that accurate, we do know that quite complicated prokaryotes were present on Earth, because that's where we find them in the fossils at that date. The first evidence of eukaryotic cells appears, we think, about 2 billion years ago. Confidently, we know that the first eukaryotic cells appear in the fossil record about 1.5 billion years ago. But there's evidence to suggest that they may have been around earlier than that, as early as 2.1 billion years ago. But the fossil evidence is not really good there. We're relying on some other ways of figuring out the origin of eukaryotic cells other than looking directly at fossils. But what that tells us is that prokaryotes were, number one, the first cell types to evolve. Number two, they've been here for the longest period of time. They dominated the landscape for over half the history of life on our planet. And while we recognize eukaryotic organisms as being, quote unquote, more dominant today than prokaryotes, the reality is there are far more prokaryotic cells on the planet in terms of numbers, in terms of biomass, the sheer mass of prokaryotic cells, than there are eukaryotes. Of all the cell types that have been most successful, it's the prokaryotes, hands down, that have been the most successful. They occupy the greatest diversity of environments. They are more flexible chemically. They are able to adapt to all sorts of weird and bizarre environments quite easily, and utilize environments that eukaryotes simply could not begin to utilize. And that's the reason why when we look for evidence of life elsewhere, we're looking for the presence of prokaryotic cells, not eukaryotic cells. Because if any cell type is likely to have evolved to utilize weird and wonderful landscapes, it's going to be prokaryotic cells from our experience. So this is, again, another table that comes straight out of the textbook, and this summarizes all the similarities and differences between prokaryotic cells and eukaryotic cells. So again, you're going to find this in the downloadable set of slides. It's also in your textbook. This is something that I would strongly encourage you to pay attention to. So we've talked about these notions that the eukaryotic cell has internal compartments called organelles enclosed by membranes, the nucleus being the most obvious and the one that is clear in when you look inside, look under the light microscope. Prokaryotic cell lacks a nucleus. One of the things that we do know about prokaryotic cells in terms of their structural features is that they have a cell wall around the outside of them. This is to stop them from swelling up and bursting because they live in an environment that's essentially pure water. Not quite pure, but it is close. And as a consequence, water has a tendency to flow into the cell. We'll talk more about that in an upcoming chapter as to why. And so in order to protect themselves and stop themselves from swelling up and bursting, they have this rigid cell wall around the outside. And as we're going to discover, that's a very common way of preventing yourself from swelling up and bursting. In fact, our cells, animal cells, are typically the odd ones out in not having a cell wall around the outside. Other things that you're likely to see around associated with prokaryotes are these short little structures called pili. Another name for them is fimbriae. These are used so that they can stick on surfaces. So you know that when you stop brushing your teeth for a day or two, material starts to build up. We call it plaque or plaque. It is, in fact, begun by some of the bacteria in your mouth using their fimbriae or their pili to stick to the surface of your teeth. And that attracts other material to stick where the bacteria are stuck, and it creates something that we call in biology a biofilm, but dentists call plaque or plaque, however you want to pronounce it. Another way of sticking to the surface is through something called the capsule, which lies outside the cell wall itself, and that can allow them to stick to a surface as well because it is sticky material. Then about 50% of all prokaryotic cells, or particularly bacteria, are motile, and they are motile, that means they can swim, because they have flagella. These are flagella that are very different from the flagella that we're going to see in eukaryotic cells, but they do allow the bacteria to move through their environment. It's a little bit like us swimming through molasses for bacteria to swim through water. So you can imagine that these structures, which work very much like a boat propeller, are generating a huge amount of energy in order for the bacteria to swim through their environment. And it allows them to encounter all sorts of molecules that they like or don't like, and they'll respond to that in a number of different ways. We don't have time to talk about it, but it's, again, a fascinating area of research that I was actually involved in for a while. When I first came to the United States, I spent some time investigating how bacteria respond to their environment, because I wanted to learn some new techniques, and this was a way that I could do so. So here you can see some of those features. So we've got the pili or fimbriae. Then you've got the capsule, that sticky coating on the outside. Underneath that is the cell wall, and then underneath that is the plasma membrane or cell membrane. Inside that is the cytoplasm or cytosol. Embedded in that are the ribosomes and a region called the nucleoid that contains the bacterial DNA, which is a small circular DNA molecule compared to what we find in eukaryotic cells. And then we have the flagella that are embedded in the surface structures of the cell. So they're anchored in the cell membrane, but also anchored into the cell wall and the capsule. At the end, and you can't see this very clearly, but at the end of the flagella embedded in this structure is in the cell wall structure and cell membrane structure, is a feature that functions like an electric motor that spins the flagellum. And it is powered by a flow of hydrogen ions. So there's the electrical charge. It really does work like an electric motor. And this is what I did research on, this little electric motor attached to the end of the flagella. It's a fascinating structure. It's made out of about 20 proteins, and it really is, to all intents and purposes, a biological electric motor. That's the best way to understand it. And it provides the energy to allow the bacteria to swim through their environment. So these are some structural features that you'll notice with a prokaryotic cell. And those are the ones that you need to know. So here's our first break, our first concept check for this presentation. So my first question is, what are the 4 structures that are universal to all cells, both prokaryotic and eukaryotic? So if you need to get a drink or take a bathroom break, grab a snack, or you want to just walk away for a little while. Now is the perfect time to do it. Otherwise, stick around for a few seconds, maybe 30 seconds, and then we'll talk about the answer to the question. So cue the Jeopardy music. So I don't know about you, but when I say cue the Jeopardy music, I can hear it playing in my head immediately. So I just run through that and then we come back. So thinking about this question, what are the 4 structures that are universal to all cells, both prokaryotic and eukaryotic? Let me change over to black for, let me see if I can pull this up. And then we'll go back. So what are the 4 structures? Well, number 1 is a cell membrane. Remember, that can also be called the plasma membrane. Number 2 is the cytoplasm, otherwise known as the cytosol. Number 3 is DNA, as the genetic material. And number 4, ribosomes. So if you had those on your list, congratulations, you know the answer to that question. And you have started mastering the structure of cells. So let's move on. So now that we've looked at prokaryotic cells, let's do a quick look at a eukaryotic cell so that we can see the similarities and differences between the 2 major types of eukaryotic cells that are our focus this semester, the plant cell and the animal cell. So again, we are going to find the presence of a nucleus. Remember, that is the defining feature of a eukaryotic cell. And there is going to be a plasma membrane. Every cell's got to have one. And then there's going to be cytoplasm that forms the material, or cytosol that forms the material in which all the organelles, including the nucleus, are going to be embedded. And that's surrounded by the plasma membrane or the cell membrane. Then we are going to find a number of other membrane-bound organelles, not just the nucleus, but we're going to find other organelles that are also surrounded by membranes. So a membrane-bound structure that's unique to plants would be the chloroplast. We know that's where photosynthesis occurs. And so we would expect the chloroplast as an organelle to be unique to plant cells, and that's what we find. We find in animal cells that there are unique organelles as well, one of which would be a lysosome, and that's involved in digestive processes. So what other structural features might we expect to find in plant cells that are unique to plant cells? Because this is something that we need to know. So plant cells also have cell walls. And those cell walls contain holes called plasmodesmata that allows plant cells to communicate with each other. So here are 3 differences that we would expect to find in plant cells. They're going to have chloroplast. They're going to have cell walls, something that animal cells most definitely do not have. They're going to have plasmodesmata as communicating channels between plant cells. These are holes in the cell walls. And there is one final feature called the central vacuole. And I'm going to qualify that by saying large central vacuole. And we'll talk more about what that does when we focus on the plant cell in a little bit more detail. When we start thinking about unique features in animal cells, well, we've got lysosomes in there. Another thing that we could put in is cilia and flagella, because this is something that you're not going to see on plant cells, but you will see on animal cells. The lysosomes are already mentioned. You can talk about something called intermediate filaments which are components of a feature that we'll talk about a little bit later in this presentation called the cytoskeleton. And then you could put in here something called centrioles, which are associated with the cytoskeleton and are associated with cell division, and they are found uniquely in animal cells, because they are also derived from a feature that's associated with cilia and flagella. And there's a whole story that we could talk about that relationship, but we're not going to get into too much detail in this class. If you ever wind up taking the General Biology 1 class, then you will get into a little bit more detail about that relationship, because it becomes something that you really do need to know for that class. But in this class, we're not going to dig deep into that, that relationship. But notice we're already starting to recognize that there are some differences between plant cells and animal cells. But are there some similarities as well? And you would expect the answer to be, of course, yes, there must be some similarities, because they are, after all, eukaryotic cells. So one of the similarities must be that they've got a nucleus. And so you can see that there is a nucleus present in both the animal cell and the plant cell. Inside the nucleus, we find the DNA, and we also find a structure called the nucleolus, which is responsible for producing the elements that are assembled to make ribosomes. We find that there are ribosomes. Again, we know that those are universal to all cells, so not surprisingly, we find that there. We find cytoplasm, or the cytosol. Again, not surprising that that's there, because that's universal. And a plasma membrane, also universal. But then there are other features that are universal, or that are common to plant cells and animal cells, that are not found in prokaryotic cells. So we have the sheets of membrane called the endoplasmic reticulum. So some of it is sheet-like, some of it is tube-like. It's all lumped together as endoplasmic reticulum. It is itself continuous with what's called the nuclear envelope, the double membrane system that surrounds the nucleus. So this is one large membrane complex. And there are ribosomes attached to the flat sheet-like part of the endoplasmic reticulum. So it's referred to as rough, because when you see it in the electron microscope, you can see the ribosomes on the surface, and it gives it a rough appearance. So it's called rough ER. The tube-like stuff is called smooth ER, because there are no ribosomes attached to it. And these have roles in manufacturing, as far as the cell is concerned, as you'll see. Then other things that you're going to find, we've talked about the ribosomes, are Golgi apparatus, or Golgi body, discovered by an Italian microscopist named Golgi, hence the name Golgi apparatus, Golgi body, because these were things that were discovered by this man named Golgi. And you can see that it looks a little bit like the rough endoplasmic reticulum, and the two are connected together by another common feature called transport vesicles, which we'll talk about in more detail as we work our way through the structural features of, the features of cells. Then you'll notice that mitochondria are common between animal cells and plant cells. In fact, mitochondria are common to all eukaryotic cells. And it's really important to remember that, because one of the common mistakes that freshman biology students make, whether you're taking the majors class or the non-majors class, is to think that plant cells have chloroplasts, animal cells have mitochondria. And nothing could be further from the truth. It is true that plant cells have chloroplasts specifically, but all eukaryotic cells, and I want to emphasize that, all eukaryotic cells have mitochondria. Very, very important to realize. So all eukaryotic cells have mitochondria. And there are even some eukaryotic cells where we can't find evidence of functional mitochondria anymore, but we know they had them because one of the things that has occurred or that we know here, is that some of the genes that are responsible for mitochondrial activity are actually found in the nucleus of cells, of eukaryotic cells. And in the eukaryotic cells where we see no evidence of functional mitochondria, there are still the genes related to mitochondria in the nuclear DNA, which tells us that while they may not have functional mitochondria now, they had them in the past because the DNA is still present. So this is why we say all eukaryotic cells have mitochondria, or have had mitochondria in the case of those cells that don't anymore. But they're parasites, so they get their energy from elsewhere, from their hosts. Plant cells have chloroplasts. And mitochondria. So just to reinforce that idea again, that mitochondria are universal to all eukaryotic cells. It's really important that you remember that, because it is a common mistake that freshman biology students make. So just to remind you again, the differences: the presence of the central vacuole, the presence of the chloroplast, the presence of the cell wall, the presence of the plasmodesmata that provide connections between plant cells. Those are all the unique features of plant cells. When we come to look at animal cells, it's the presence of the centrioles, the presence of the lysosome. We can also say the presence of cilia and flagella, and one component of the cytoskeleton that is found in animal cells and not particularly found in plant cells. So there are a lot of similarities. There are some differences, and it's very helpful. I found this to be very helpful for students. If you create a table with 3 columns, head the center column, common features, head one of the other columns, unique to plant, and the third column, unique to animal. And then put things into each of the columns based on whether they are common to plant and animal cells, or they're unique to a plant cell or unique to an animal cell. It helps you figure out which things are found in both types of cells, which are found uniquely in one type of cell versus the other type of cell. And then double check to come back to this picture, come back to the textbook and double check to see if you've got everything in the right columns. If you haven't, put it away, come back and repeat the exercise a little bit later in the day or the next day. And again, compare, make sure that you correct your errors every time that you do this until you can write out all the things that are in common and all the things that are unique to each cell type from memory. Because then you can say, I know this, I've got it, I know this. It's a very, very helpful way of learning all of this. And it is something that you do need to know, by the way. So we also have 2 animations from Pearson. One is a 3.5 minute trip through a typical animal cell. The second one is about a 3.5 minute trip through a typical plant cell. And again, it reinforces the similarities and the differences, and it actually shows you the features in each type of cell. And again, remember, you can find these as links in the resource page, and this is chapter 4, so this will be in the chapter 4 resource page within the week 4 module for this class as it exists right now. So if you want to look at these animations and get a 3.5 minute tour through each cell type, that's where you would want to go. You can also find these in the study area in Mastering Biology for chapter 4 if you haven't looked there. So there are a number of different ways that you can look at these animations. So time for another quick break. Probably unless you didn't take a bathroom break or a drink break at the last break, you're probably good to go at this point. So let's take a look at the concept check. Take 30 seconds to think about it, and then let's take a look at the answer to the question. So name the structures that are unique to animal cells and plant cells respectively. We just talked about this, so this should not be very difficult, but cue the Jeopardy music. Okay, let's see what we've got. If we're talking animal cells, remember we're talking lysosomes, we're talking centrioles, not mentioned in the diagram there, but we're also talking cilia and flagella. And we could put in there as well intermediate filaments as components of the cytoskeleton. For plant cells, well, we're talking the chloroplast, we're talking the central vacuole, the cell wall. And while they're not specifically named, they're just called communicating channels on the diagram that we were just looking at. We could put in there the plasmodesmata as well as to have the name of the communicating channels between plant cells. So if you've got a list that looks somewhat like mine, congratulations. You're starting to master the structural features of typical eukaryotic cells. So time to move on again. So the first feature that we need to focus on is the plasma membrane. We're going to talk a little bit about the structural nature of the plasma membrane at this point, but in an upcoming chapter, we're going to talk about its functional features. So there are two things that we need to bear in mind. Number one, the basic nature of the membrane, and it doesn't matter which membrane we're talking about. It could be the plasma membrane, it could be the membranes associated with different organelles. The basic structure of a cell membrane is a bilayer of phospholipids. Now, you may recall from a previous chapter that a phospholipid is an amphipathic molecule, meaning it has a hydrophobic region and a hydrophilic region. The hydrophobic region is represented by the fatty acids that are a part of the lipid structure, and the hydrophilic region is represented by the attached phosphate group and another organic group that is a non-fatty acid group that is also charged. So you've got a charged region and an uncharged region. So when you mix that with water, it spontaneously organizes itself into this phospholipid bilayer, with the very hydrophobic fatty acids pointing in towards each other and the very hydrophilic region of the phosphate group and that other organic group pointing out to the water on either side of the bilayer. And this is the basic form of every cell membrane. It forms a very, very nice barrier between the water on one side of the cell, which could be the outside of the cell, and the water on the other side, which could be the cytoplasm of the cell. Remember, most of the content of a cell is water, about 70% in most cases. So the cytoplasm is basically a solution with water, and the outside of the cell is most definitely, for most cells, water as well. And so you have this very hydrophobic region in the center and the hydrophilic on both sides. And so this is a spontaneous structure that forms. Then embedded in the phospholipid bilayer are proteins. You have both integral proteins that are embedded in the membrane, and these, as you might guess, are characterized by having hydrophobic regions that allow them to interact with the hydrophobic regions of the phospholipids. But they also have hydrophilic regions that point outwards into the water surrounding the cell or into the water solution that is the cytoplasm. And then on the surface of the membrane, you have a set of proteins that are referred to as peripheral. And these are hydrophilic because they're associated with the water solution that is the cytoplasm, or they could be on the outside surface of the cell, associated with the water environment that is outside of the cell. So you've got these two basic types of proteins. You've got the integrated proteins or integral proteins that are embedded in the membrane itself. And then you've got the peripheral proteins that are usually on the surface of the membrane. They are hydrophilic, and they're carrying out functions as well. And what makes the difference between different types of membranes is not the phospholipids, because a phospholipid bilayer is just that, a phospholipid bilayer. What makes the difference is the types of proteins that are associated with each membrane. So when we start thinking about membrane function, we have to start thinking about the types of proteins that are buried in the phospholipid bilayer. So when we talk about the function of membranes in an upcoming chapter, we're going to focus a lot on the types of proteins that are present, because that's really going to tell us a lot about what the function of the protein of the membrane is. What can it actually do for the cell? So sometimes membrane proteins, or proteins that are associated with membranes, can serve as a passageway for disease. So there is a type of bacterium, we sometimes refer to it as golden staph, or methicillin-resistant Staphylococcus aureus, or multiply resistant Staphylococcus aureus. All those names apply to the same type of Staphylococcus aureus. It's a type of bacterium, and it's bad news because most of our common antibiotics simply don't work against it anymore. And so if you become infected with MRSA, which is the way we pronounce this, it can be really life-threatening because most of the common antibiotics are no longer working against it. And the question has become, is it possible that there are different types of MRSA that are less virulent, less deadly, than the more common strain of MRSA that is the one that typically winds up killing people? And we know that the really deadly strain of MRSA has this protein called PSM. And it's thought to interact with proteins on the surface of human cells and disrupt those in such a way that it makes us vulnerable to other diseases because it affects human immune cells. Sounds kind of familiar, doesn't it? Because we know of other things that do this, like the HIV virus. And from history, the plague bacterium called Yersinia pestis, which used very much a similar mechanism to HIV, the one that HIV uses. So MRSA also is known to interfere with the immune system. Is it PSM that makes the common form of MRSA as deadly as it is and makes MRSA and all strains of MRSA as deadly as they are? So it turns out that MRSA is just as deadly to mice as it is to humans. So the experimentation was done in mice, and mice were infected with both normal MRSA that makes PSM and one of these quote unquote less deadly versions of MRSA that doesn't make PSM. And we did, in the results for the experimentation, all the mice that were infected with normal MRSA making PSM did die. So it is very lethal. And the mutant version of MRSA that doesn't make PSM was deadly, but obviously not as deadly because not all the mice died. A few of them survived. But what that tells us is that even in the absence of PSM, MRSA can still kill. So there must be other things that are also contributing to the lethal nature of MRSA. It's not just PSM. PSM is sort of the final nail in the coffin, so to speak. It's what makes normal MRSA as deadly as it is. But there are other things going on as well, other than the presence of PSM, that makes MRSA almost as deadly as it would be if the PSM was present. So you can't just say, well, if we can knock out the strain of MRSA that makes PSM, the other strains are not going to hurt us. That's clearly not the case. MRSA, in its many forms, is deadly. It doesn't matter whether it makes PSM or not. It is deadly, and if you happen to be susceptible to one of the non-PSM making strains of MRSA, it's going to kill you just as easily as the normal MRSA will. That's the upshot of this research. So when we start looking at the surface of cells, so we've got this membrane around the outside of the cell, remember every cell's got one, and we saw that outside of a bacterial cell there are cell walls. Well, we know that outside of a plant cell there's also a cell wall, and it's made out of cellulose. And cellulose is the primary component of all plant cell walls. It's also the major component of what we refer to as wood. Why? Because wood is plant cell walls. So what we call wood, what we cut out of a tree that we refer to as wood, is basically the leftover cell walls after the plant cell itself inside the cell walls dies. And so it is the presence of the cellulose, and we talked a little bit about cellulose when we talked about the macromolecules of cells. It's the cellulose that gives wood its strength. And it's not a bad analogy to say that a plant cell is a cell inside a wooden box, because remember, wood is the cell wall and the plant cell is sitting inside that cell wall structure. So it's really a cell inside a wooden box. And it's what stops a plant cell from swelling up and bursting. But the plant cell does swell until it's pushing hard against the wooden box, the cell wall, which of course pushes back and prevents the cell from swelling any further. And it's a way for plant cells to regulate the flow of water into them. When you forget to water your houseplants, and we've all done this, right? And you see the plant starting to wilt, what that tells you is there's not enough water pressure inside the plant cells to keep the plant fully erect anymore. And you can fix that really quickly. You just water the pot, the plants take up the water, and the water pressure is restored. We refer to it as turgid. And it maintains the rigidity of the leaves and the stems and allows our little soft houseplants to stand upright, because they don't have as much supportive material as, say, a tree or a shrub with its wood, its woody nature on the inside. But that water pressure is still present. Animal cells don't have cell walls, but they do have something called an extracellular matrix, which is a complex environment of fibers made out of complex sugars and proteins. There are lots of proteins that stick out of the plasma membrane into the environment and connect to the extracellular matrix. It allows for communication between animal cells, but the one thing that it isn't is a rigid structure like the cell wall around a plant cell, or the cell wall around a fungal cell, or the cell wall around around bacterial cells. In fact, animal cells are the odd ones out in not having a rigid cell wall around the outside to stop them from swelling up and bursting. And when we talk in more detail about the functional nature of membranes, we'll talk more about how cells regulate the flow of water into and out of them, and we have to focus with very sharp focus on animal cells because they do things differently. If they don't have cell walls, then they must have a different way of regulating the flow of water. So again, when we recognize that there is no connection or no rigid structure around cell walls, then there must be other ways of animal cells communicating with each other. So there are, in terms of connections, so we have things called tight junctions. And what's really important about these is that they are watertight. And a good example is in the skin. Then we've got something called desmosomes. And these anchor the cells to each other. And a really good example of this is muscle cells. In muscles, these are what you break when you tear a muscle. This is why a muscle tear hurts so much, because you're ripping open these connections between the muscle cells, and it really hurts. And then we have something called gap junctions. And these are for communication. And a really good example of these is in between nerve cells, where there is a definite gap between the adjacent nerve cells, and it requires chemicals to be released from one nerve cell and be picked up by the next nerve cell in order for the nerve impulse to be transmitted. We don't need to go into the details of this. I just wanted to let you know that there are connections between animal cells in the same way as there are connections between plant cells, but they are of different types because of the different nature of plant cells and animal cells. So, quick concept check before we move on. Why do phospholipids spontaneously form a lipid bilayer when mixed with water? And of course, the answer here is they are, you want to use the correct term, amphipathic. Hopefully you remember that means they have both a hydrophobic and hydrophilic region. The hydrophobic fatty acids come together to exclude water. And the hydrophobic heads point out. So if this is the thought that came up, again, congratulations. You're starting to master the nature of membranes. Remember, we are going to talk in way more detail about membranes in an upcoming chapter. But this is where you start to master the notion of membranes. So once we start thinking in terms of the internal membrane-bound organelles, the internal compartments, there are a number that immediately come to mind. The first, of course, is the nucleus. And here you can see an artist's diagram of a nucleus. And you can see that the nuclear envelope is surrounded by a double membrane. So this is two lipid bilayer structures penetrated by protein complexes called nuclear pores. And you can see an electron microscope image of nuclear pores here buried in the nuclear envelope. And these are the gatekeepers. They regulate what can move into and out of the nucleus. Because you don't want just anything getting into or out of the nucleus. You do need to be able to regulate that. And then inside the nucleus, we're going to find the DNA in the form of chromatin. We talked about that before. That's the normal form of the DNA inside the nucleus. We only recognize chromosomes when the DNA condenses into more visible structures that are present when the cell is actually dividing, but under normal circumstances, it's in the chromatin form. You also have, and I mentioned this very briefly when we were talking about animal and plant cells, the presence of a nucleolus. There could be one or more of these inside the nucleus, but this is where the components of ribosomes are produced and released out into the cytoplasm to be assembled into the ribosome structures that are going to assemble polypeptides. So this is what we see inside the nucleus. This is a classic nucleus structure. And I've talked about the various features. So here is the DNA in its chromatin form. It's around some proteins called histones. These are unique to eukaryotic cells. We don't find histones anywhere else. And they create complexes that the DNA wraps around, and then the DNA itself spirals into a fiber core, which becomes the chromatin fiber. At the point of cell division, that then coils up and gets folded up even further in a process called condensation that gives rise to the chromosome structure that we see during cell division. So this process is called supercoiling. So the supercoiling occurs when the cell is prepping for cell division. And so at the beginning of cell division, you actually have visible chromosomes because they've become supercoiled. They've gone from the chromatin form to this supercoiled chromosome form as part of the prep for cell division. Then embedded in the cytoplasm are the ribosomes that are responsible for the assembly of polypeptides. They take the information of a messenger RNA molecule, and we'll talk a little bit more about that as we progress through this section, and use the information in that molecule, the messenger RNA molecule, to assemble amino acids into polypeptides. So the ribosomes themselves are out here in the cytoplasm. These are the little black dots. All ribosomes start out as free ribosomes. what happens next depends on the type of polypeptide that's being made. So we're going to talk about a system called the endomembrane system, which specializes in making polypeptides and proteins that are ultimately either going to go to the plasma membrane and become part of the proteins found in the plasma membrane, or they're going to be released from the cell itself into the cell environment. So if those if the ribosome is making polypeptides that are going to enter the endomembrane system, then the ribosomes move from the cytoplasm down to the surface of the rough endoplasmic reticulum. And all of these little black dots that you can see attached to the surface of the rough endoplasmic reticulum and make it look rough, those are actually ribosomes that are assembling polypeptides that are going to enter this endomembrane system that I've referred to. So all ribosomes begin their functional life as free ribosomes in the cytoplasm. If they're making a polypeptide that is not destined for the endomembrane system, then they'll stay in the cytoplasm. If, however, they're making a polypeptide that's destined to go into the endomembrane system, they move down to the surface of the rough endoplasmic reticulum to produce their polypeptide, because the ultimate result is that the polypeptide enters into this hollow space of the rough endoplasmic reticulum, which is referred to as the lumen. And we'll talk a little bit more about that as we progress through this section. So the ribosomes on the rough are moved on the cytoplasm to the ER. So that should help you remember that. So we've got these two populations of ribosomes, the free ribosomes that are in the cytoplasm, and then the attached ribosomes that are attached to the surface of the endoplasmic reticulum because of the polypeptide that they're generating. So how does the ribosome get the message to assemble a polypeptide? Well, this is part of a process that we're going to examine in more detail, and you're actually going to make an infographic on later in the semester, called the central dogma of the molecular biology of the cell. And it's all about information flow. And the information flow is said to be from DNA to RNA to polypeptide. And so inside the nucleus, a copy of the information on the DNA is made in the form of this RNA molecule called messenger RNA. That then leaves the nucleus and is captured by a ribosome. And the ribosome reads off the information in the messenger RNA and uses that to assemble the polypeptide or the protein. And so that is the nature of this flow of information. It is from DNA to RNA to polypeptide. The copying of the information in the DNA molecule into the messenger RNA occurs in the nucleus, but the assembly of the polypeptide or the protein takes place in the cytoplasm where the ribosomes are located. And so this is how the ribosome gets the information to assemble the polypeptide. And again, remember, it's the nature of that polypeptide that determines where the ribosome is going to wind up. If the polypeptide is not intended to enter the endomembrane system, then the ribosome stays in the cytoplasm. However, if the polypeptide is intended to enter the endomembrane system, then the a ribosome moves down to the surface of the rough endoplasmic reticulum. So let's take a look at the endoplasmic reticulum next, but before we do, quick concept check. So what is the function of ribosomes and what role does the messenger RNA play in making polypeptides? So as we've just been talking about this, that should not be very difficult. What are the function of the ribosomes? To assemble amino acids into polypeptides. So that's the function of ribosomes. Messenger RNA. That contains the information that ribosomes use to correctly assemble a polypeptide. So there we have the answer to this question. Not a very difficult question to answer, but an important one to answer. So time to move on. Endoplasmic reticulum next, and the endomembrane system. So when we start thinking in terms of the endomembrane system, it is very convenient to think of this as the manufacturing plant of the cell, because in essence, that's what it does. It makes lipids. Let me put in here. So the endomembrane system makes, well, we know one thing, polypeptides. That's the rough ER. It makes sugars. I think I'll erase that. So it makes sugars. That's smooth ER. It makes lipids. Also smooth ER. It also stores calcium. And that will be smooth ER. And then it stores and packages proteins. And that's primarily the Golgi, but also the rough ER. So these are the various functions that are associated with the endomembrane system in terms of manufacturing, transporting, storage, modification. These are all things that we're going to see associated with the endomembrane system. So you can understand why this is an important system to come to grips with, because it's central to the way the cell makes things. So this diagram shows the flow of material through the endomembrane system. So you could think of this in terms of any protein that is going to turn up in the plasma membrane of the cell, or any protein that's going to be released into the cell's environment. And the classic protein that we often think about when we think about movement through the endomembrane system is the protein hormone insulin, the one that regulates glucose or blood sugar. So when there is too much glucose in the bloodstream, the cells of the pancreas, through their endomembrane system, start generating insulin, or mobilizing insulin that has been pre-made and stored in the Golgi apparatus, so that it can be released into the cell's environment, which is basically into the bloodstream, so that it can trigger cells to start taking up the glucose to reduce the glucose content of the blood system. And so, because it's going into the endomembrane system, it's going to be made by ribosomes attached to the surface of the rough endoplasmic reticulum. The polypeptide enters the endoplasmic reticulum. It gets packaged into something called a transport vesicle that buds off the surface of the rough endoplasmic reticulum and carries the polypeptides to the cell's warehouse, the Golgi apparatus. There, the transport vesicle merges with the membranes of the Golgi apparatus, and the proteins enter the Golgi apparatus. They may get stored, they may get repackaged, they may get chemically modified, but then eventually they'll move out of the Golgi apparatus by being repackaged into transport vesicles, a specialized version of which is the lysosome that contains digestive enzymes for internal digestion inside the cell, or as a transport vesicle carried to the surface of the cell, where the transport vesicle merges with the plasma membrane to release its contents to the outside of the cell. This is the endomembrane system in a nutshell. Okay, so you can see that there are multiple organelles involved here. The endoplasmic reticulum, both rough and smooth, are involved. These transporting vesicles that tie everything together, the Golgi apparatus that stores and modifies and repackages everything. And then some specialized transport vesicles that carry out specialized functions inside cells. All of this is tied to the endomembrane system. So it's a major system to come to grips with. So we have the endoplasmic reticulum, made up of smooth endoplasmic reticulum and rough endoplasmic reticulum. And remember, it is also connected to the nuclear envelope. So this is one large membrane system. Both the nuclear envelope and the rough endoplasmic reticulum have ribosomes on the surface, making polypeptides that are going to enter into the hollow space of the endoplasmic reticulum. And that's involved, and so the rough endoplasmic reticulum specializes in making polypeptides. The smooth endoplasmic reticulum makes the sugars, it makes the lipids, and it's actually making lipids that are going to be incorporated not only into the endoplasmic reticulum, but into the nuclear envelope, into the transport vesicles, into the Golgi apparatus and the lysosomes, and eventually into the plasma membrane itself, because they're all tied together by the transport vesicles that are budding off the endoplasmic reticulum and therefore carrying lipids as part of their membranes to the Golgi, where they get incorporated into the Golgi. And then eventually, when the transport vesicles and lysosomes bud off, they remove more lipid as membrane and either retain it as a lysosome or carry it to the cell membrane when the transport vesicle merges with the plasma membrane. So the smooth endoplasmic reticulum is a lipid-producing structure, really critical to all of the membranes of the cell, and there's evidence to suggest that they also contribute to the membranes of the mitochondria and the chloroplast. So these are major sources of the phospholipids that make up the membranes. So again, part of the manufacturing system of the cell, really, really important. We talked about the rough endoplasmic reticulum as having the ribosomes on the surface. So here you can see the ribosome attached to the surface. You can see the polypeptide being released into the hollow space of the rough endoplasmic reticulum, folds up into its final shape, and then gets packaged into the transport vesicle that's going to carry it either to its final destination, which is a possibility, or carry it to the Golgi if it needs to be stored. So we talked about the smooth endoplasmic reticulum making lipids. I also mentioned it makes sugars. One of the other big features of the smooth endoplasmic reticulum is that it's a detox. So in the liver, the smooth endoplasmic reticulum is responsible for breaking down any drugs that you may take, which is the reason why you need to retake them, because the liver is constantly breaking them down. And this includes alcohol. And this is the reason why if you abuse alcohol, your liver swells. So let me put in here, if you use alcohol, so this is why alcohol abuse eventually becomes lethal, because the liver's response to all of that alcohol is to swell to try and make more smooth endoplasmic reticulum in the cells to cope with all of the alcohol. The problem with that is that eventually the liver functions stop because it can't continue to do this. And remember, alcohol is ultimately a cell poison, a cell toxin. So if it can't cope with that, it does eventually wind up killing the liver. And once your liver stops functioning, you've got about 3 months to live. Because you can't live for more than about 3 months without a functional liver. The only alternative is to give the liver transplant, is to get a liver transplant, which is very, very difficult to do. It's one of the hardest transplants to organize. So something to think about. Make a connection to life as we know it, I guess. So let's talk about the Golgi apparatus. Again, it's the cell's warehouse. So when we take a look at the Golgi apparatus, there are two clear sides to the Golgi apparatus. And you can see that it looks very much like an extension of the endoplasmic reticulum in terms of the folded sheets of membrane that look very much like the rough endoplasmic reticulum. So given that it's a warehouse, most warehouses have an input side and an export side. And the Golgi apparatus is exactly the same. And so one side of the Golgi faces inwards towards the nucleus, and this is the receiving side, always also known as the cis side. Oops. Let's neaten this whole thing up. Hold that. And you may recall from when we talked about the fatty acids, we had the cis and the fat and the trans fatty acids, or the cis and the trans geometric isomers. So that's where the prefix means the same thing, same side or opposite side. In this case, same side. So it's facing the nucleus. The other side, or the shipping side, is called the So the shipping side is sometimes referred to as the trans side, meaning it's on the opposite side to the nucleus, and it faces out towards the plasma membrane. So when the transport vesicles bring the polypeptides from the rough endoplasmic reticulum, its membranes merge with the membranes of the Golgi apparatus. The proteins and polypeptides then enter the Golgi apparatus and passage through. They may get stored for a period. They may get chemically modified. They may get repackaged with other polypeptides to form proteins, but eventually they're going to get packaged into another transport vesicle, which is going to then carry them to their final destination, which could be somewhere inside the cell, or in the case of most of them, out to the plasma membrane, where they'll merge with the plasma membrane and release the polypeptides out into the environment. Or if they're polypeptides that are inserted into the membrane, they'll become part of the protein complement of the plasma membrane itself. So this is the cell's warehouse. It stores, it ships, it repackages, it chemically modifies. That's the role of the Golgi. The lysosomes then are a specialized type of transport vesicle. Their job is to not so much carry things to a final destination inside the cell. It's more to connect up with either other vacuoles or vesicles that contain food particles, or that contain worn out organelles. Their job is to digest things inside the cell. So they contain enzymes that are digestive, and they will break down all the macromolecules. So an example of lysosomes at work is the loss of the tail as a tadpole becomes a frog. So what's happening here is that the lysosomes inside all the cells that make up the tail of the tadpole break open and release their digestive enzymes, and those enzymes go to work and self-digest. They literally digest the cells that they're in. It's a process called autophagy, where the cell digests itself. And in this case, it's programmed. It's an intentional programming step in the life of the tadpole, because a frog doesn't need a tail. Tadpole does, frog does not. So rather than waste all the good molecules in the tail of the tadpole, the lysosomes break open, and the molecules are recycled back into the body of the frog to be turned into other structures that are beneficial to the frog, as the metamorphosis of the tadpole to a frog takes place. So this would be a good example of the role of a lysosome that you would be familiar with. So we also know that they're responsible for breaking down food particles in some protists that feed this way. So this would be amoebas. cells called phagocytes. And these are actually white blood cells. Destroy bacteria. So after they capture the bacteria, and we'll talk in an upcoming chapter about how they do that, they actually use the lysosomes then to destroy the bacteria. Often it winds up killing the phagocytes. So if you've ever seen pus associated with a cut, then you've seen the dead phagocytes that have been eating the bacteria, and as a consequence have been killed by the toxic substances that are released by the dead bacteria. Better that these cells die than the whole organism, you, die as a consequence of the bacteria. So it's mentioned there as well. Oh, yeah, during our own development, our hands and feet start out as paddles and you can't actually see the fingers and toes. But the skin between the fingers and toes die as a process called necrosis. And again, this is the lysosomes at work. I forgot that this was mentioned as an example. So this would be something that would be would relate to do, but the tadpole example is just as relevant as well. It's the same thing, same idea. So here you can see the two different ways of dealing with of the lysosomes at work. Here it is breaking down food particles, and here it is getting rid of worn out organelles inside a cell. And there are actually a number of diseases that are associated with dysfunctional lysosomes. And the problem with this is that these are diseases that are inherited, and there is typically no cure for these. They can be quite devastating in a family when it's understood that it was passed from parent to child. And very often, the kids die very young, and there's nothing you can do about it. There's no treatment for it. You just have to sit and watch and wait for the inevitable to happen. And for the parents, I just can't imagine how devastating it must be, because it's an inherited disease. So it's the parents that pass this on to their kids. I just can't begin to imagine how devastating it must be to have a child with this condition and know that there's nothing that you can do about it, but it was you that passed the genes on to your child, and that's the reason why they have it. We're going to talk about a number of these sorts of genetically determined diseases as we move deeper into the semester in some upcoming chapters. Vacuoles have a number of different functions. So in animal cells, we have the food vacuoles, and in protists, we have food vacuoles that allow for the cells to feed. In some types of protists, and this is paramecium, you have a specialized vacuole called a contractile vacuole that actually functions as a water pump. It allows the cells to capture water that's flowing in and pump it back out again, as a way of regulating water flow so that they don't swell up and burst. It requires energy, but it's an energy investment that is beneficial to the cell, because if it didn't do this, it would swell up and burst, and that would be lethal. So investing some energy in pumping the water out, great investment as far as these cells are concerned. In plant cells, you have the central vacuole, and you can see how it dominates the internal space of the cell. This is the nucleus, by the way, very small compared to the central vacuole. And it's the accumulation of water in the central vacuole that causes the plant cell to swell and creates that turgid condition that is typical of all plant cells. The reason why leaves are expanded, our houseplants stand upright, is because of the accumulated water in the central vacuole. When that water content decreases because the plants are not getting enough water, that's when the pressure is relaxed and the plant starts to wilt. And of course, you can restore the normal condition by simply watering the plants again. Another feature of the central vacuole, so I'm going to put in here, accumulates water. Stores waste. And can store So one thing to remember about plants is that they don't have a circulatory system or a waste disposal system like animals. So when they produce toxic waste, they can't get rid of it easily. So the way they deal with that is to store it in the central vacuole away from the cellular chemistry. But it can also store beneficial molecules. So if you had a glass of orange juice this morning for your breakfast, then you are drinking vacuole juice. The vacuole juice is actually a solution of sugars and other materials that the orange specifically stores there so that you will want to eat it. Why? Because the orange is a fruit and it typically contains seeds. Of course, navel oranges don't, but they're a mutation. But a orange tree wants you to eat the orange because hopefully you'll consume the seeds and they'll pass through your digestive tract and get transported to another location to spread the trees over a greater distance. That's why plants make fruits, and that's why fruits have all sorts of good things in them that make them taste good, that make them smell good, that make them look good. But really, they're all about getting the seeds moved from the parent plant to another location in the gut of the animal that ate the fruit. And so when you eat an orange, you're doing exactly what the orange tree wants you to do. Of course, drinking a glass of orange juice gets around that, but that's another story for another day. So, quick concept check so that we can continue moving on. This one's a little bit more complicated, but it's an important one to think about, because if we're going to make sense of the endomembrane system, it's really important to remember what the passage of a polypeptide is through the system. So the protein hormone insulin is produced in cells in the pancreas. List the organelles of the endomembrane system in the correct order that would be involved in this process. So cue the Jeopardy music for about 30 seconds. So let's think about what the organelles in sequence might be. Well, first of all, we've got the nucleus, because that's where the DNA is located, that's where the genes are located. So we've got to make a copy of the gene in the form of the messenger RNA. The next organelle would be ribosomes. And if you wanted to be technically correct, it would be a free ribosome, which then moves down to the surface of the rough ER so that the polypeptide can be inserted into the hollow space of the ER, what is referred to as the lumen. That's the fancy name. From the rough ER, then it would be transport vesicles. which would carry it to the Golgi. There it would be modified, repackaged into another transport vesicle. Which would carry it to its ultimate destination, which is the plasma membrane. So hopefully those are the organelles that you had on your list, and that's the order that you had them in. And if you did, then congratulations, because you're starting to master the endomembrane system. So onwards and upwards to the last major group of organelles. Yes, well, getting there a little bit slower than I would like, but we're getting there. Actually, we've got two more sets. I misspoke. So the next group of organelles are very easy to understand. These are the energy processing organelles. And there are two major organelles that are involved here. There is a third one, but we're only going to talk about two. And that's the chloroplasts and the mitochondria. So chloroplasts, remember, do photosynthesis. And the mitochondria are involved in the aerobic part of cell respiration. And remember again, while it is true that only plant cells have chloroplasts, it is not true that only animal cells have mitochondria. All eukaryotic cells have mitochondria, which means that all eukaryotic cells are potentially capable of aerobic cellular respiration. Whether they do it or not, that's going to be the subject of an upcoming chapter when we talk about cell respiration. So we'll save that for another day. So chloroplasts carry out photosynthesis. When we look at the structure of a chloroplast, it reflects this function. So the first thing to recognize is that the chloroplast, like the mitochondria, is surrounded by two membranes. They are not connected to each other, so they are separate from each other. So this makes it a little bit different from the double membrane system of the nuclear envelope. The inner membrane is connected to the third membrane system of the chloroplast, this highly folded series of disc-like structures called thylakoids that are arranged into stacks called a granum, plural grana. Each of these stacks is in turn connected to adjacent stacks by these connecting membranes, sometimes referred to as lamellae membranes. What the important point here is to remember is that the internal space of all the thylakoids in each granum are connected together. And then the internal spaces of each granum is connected to the internal spaces of the adjacent grana by these connecting membranes, creating one large internal space called the thylakoid space. It is separated from a second compartment in a chloroplast, what is the functional equivalent of the cytoplasm of a cell called the stroma. So what you've got is two separate compartments, the stroma and the internal space of the thylakoid called the thylakoid space, separated by a membrane. And that's really important because that is required to carry out photosynthesis. So why have all of these folded membranes? To maximize your ability to capture light energy. And light energy is captured by complexes embedded in the thylakoid membranes. And that light energy is then converted to chemical energy by other protein complexes embedded in the thylakoid membranes. That energy is then used by enzymes in the stroma to convert water and carbon dioxide into organic molecules. So you start to see how the structure of the chloroplast reflects its function as a light capturing organelle that uses the light energy to store chemical energy in the form of organic molecules. A lot more to come in an upcoming chapter. Mitochondria also have a reasonably complicated internal structure, and that internal structure reflects its function as generators of ATP, the endpoint of cellular respiration and the major energy molecule or energy currency of cells. So like chloroplasts, they are also surrounded by a double membrane system. The outer membrane system is not connected to the inner membrane, but the inner membrane is highly folded into fingers called cristae. So this inner membrane is sometimes referred to as a cristae membrane. Again, there are two compartments here. The first compartment is equivalent to the stroma of the chloroplast. It's referred to, however, as the matrix, and it is functionally equivalent to the cytoplasm of the cell. The second compartment is the space in between the inner and outer membrane, which is referred to as the intermembrane space. And these two compartments are, of course, separated by the cristae membrane, the folded inner membrane system. And this is central to making ATP. The folding occurs because if you fold the membrane, you have a lot more membrane surface area. And that tells you that that folded membrane must be associated with the function of the mitochondria. Otherwise, why bother folding it? And that's what we find. The production of ATP is associated with the cristae membranes. So having more membrane means the ability to make more ATP. And so all of the protein complexes that are associated with making ATP, we're going to find those in the cristae membrane. We're also going to find that there are a number of enzymes in the matrix that help in the breakdown of molecules derived from glucose, the primary fuel for cellular respiration. And you're going to see that there's going to be a lot of tie-in again between the structure of the mitochondria and the ability to carry out the aerobic portion of cell respiration. So more to come in the upcoming chapter devoted to cell respiration. What is interesting about mitochondria and chloroplasts is that they seem to have a lot of features that are very bacterial in form. They have their own DNA, and it's circular DNA, like bacterial DNA. They have their own ribosomes, and the ribosomes of chloroplasts and mitochondria are of the same type as bacterial ribosomes. And they have the same properties. There are specific antibiotics that shut down the ribosomes of bacteria, and they also shut down the ribosomes of mitochondria and chloroplasts so that they can't make proteins. The mitochondria and chloroplasts do actually make some of their own proteins. As I mentioned, some of the genes of mitochondria are found in the nucleus now, and some of the genes associated with chloroplasts are also found in the nucleus of plant cells. But they do have this semi-independent behavior, the semi-independent of the cells that they're living in. And in fact, when the plant cell or animal cell divide, the chloroplasts and mitochondria divide separately in a separate process that is very much like bacterial cell division. If all of this starts to make you think that chloroplasts and mitochondria sound very bacterial-like, then good, because that's what we think they once were. Way back in deep time, a precursor to eukaryotic, all eukaryotic cells captured mitochondrion, but rather than, or captured an aerobic bacterium. But rather than turning it into a food source, it kept it, because it turns out that that was a benefit to the eukaryotic cell, because the bacterium was turning out boatloads of ATP, and that was of benefit to the eukaryotic cell. And so they came to an arrangement which is called a symbiotic arrangement. We actually refer to this as the endosymbiotic theory of the origin of mitochondria and chloroplasts. That both of these organelles were once free-swimming bacteria, in the case of the mitochondria, an aerobic bacterium, in the case of the chloroplast, a cyanobacterium carrying out photosynthesis. The first to get captured was an aerobic bacterium because all eukaryotic cells contain mitochondria. So it must have been very early on in the evolution of a eukaryotic cell that the aerobic bacterium that would become the mitochondria got captured. At a later point in time, a cyanobacterium was captured by a eukaryotic cell that already had a mitochondrion, and that would eventually become the chloroplast. And remember, only plant cells have chloroplasts. All eukaryotic cells have mitochondria, so it must have occurred in that order. And this idea was first floated back in the 1970s by a biologist named Lynn Margulies. And it took a while for people to become convinced that Margulies was correct. It would take nearly 20 years for the evidence to accumulate that Margulies was correct. But today, we know that Margulies is correct, so much so that we consider this to be a theory. This is not a hypothesis anymore. This is a theory. And it is the best explanation that we have of the origin of chloroplasts and mitochondria. Because to come up with another way for these to evolve is just beyond imagination. It was something that people really struggled with until Margulies floated this idea. And even though people were not willing immediately to accept her idea, eventually, as people started to put it to the test, which is what we always do when someone comes up with a quote unquote novel idea, it became obvious that the data that everyone was collecting was very supportive of Margulies' idea, and eventually became so obvious that this must be the origin of chloroplasts and mitochondria, that no other way of mitochondria and chloroplasts evolving makes any sort of logical sense anymore. So this is where we believe mitochondria and chloroplasts came from. We're very confident of this, that we've given it the theory tag rather than hypothesis tag. So last, I believe this is the last, or maybe it's the second last break time with a concept check. There may be one more. So here is the question for the moment. What is wrong with this statement? Plant cells have chloroplasts and animal cells have mitochondria. 30 seconds of Jeopardy music. Now, this should not be terribly difficult for you to figure out because we've just been talking about this. What's wrong with this statement? It is true that plant cells uniquely have chloroplasts, but it is not true that animal cells uniquely have mitochondria. Remember, mitochondria are universal. Even plants must do cellular respiration during the night to support themselves, because photosynthesis can only occur during the day. And while that may be the source of the organic molecules that plant cells produce and store, they still need to carry out cell respiration to break them down, to extract the energy out of them, just like every other eukaryotic cell does. So plant cells have both mitochondria and chloroplasts. They do both cell respiration and photosynthesis, both during the day, cell respiration at night. Animal cells only do cell respiration and are dependent on the molecules that plant cells are assembling during the day, during photosynthesis. So if that was the thoughts that you had, if that was the solution that you wrote down, congratulations again, because you're starting to master chloroplasts and mitochondria. So now we come to the last group of organelles, which is a catch-all group of organelles that basically involves around organelles that don't fit into the other energy processing or manufacturing group of organelles. These include the cytoskeleton and some structures that are related to the cytoskeleton, cilia and flagella. So when you hear the term skeleton, you might think of a rigid structure, but skeletons don't have to be rigid. They can be flexible, just so long as they are frameworks that supply support to the feature that they're involved in. And that is the nature of the cytoskeleton in cells. It's not rigid, it is actually quite flexible. And when we see cells, particularly eukaryotic cells, changing shape, that change in shape is driven by the cytoskeleton. So right there, we see the lack of rigidity in the cytoskeleton, but it still helps the cell maintain its shape, and it can actually, through its flexibility, allow the cell to change shape. And that is, in fact, really important, because your muscle cells could not contract to cause your muscles to contract if the skeleton was rigid. It has to be flexible in order to allow muscles to contract. And it is part of the cytoskeleton that allows that to happen. So in terms of the cytoskeleton, there are three structures called microtubules, intermediate filaments, and microfilaments. The microtubules are the largest and most clearly seen. The microfilaments are the smallest and are the elastic features that allow muscle cells to contract. And then in between, in animal cells, are some fibers called intermediate filaments that are made out of a protein called keratin, which is the same protein that makes your hair, your fingernails, claws, beaks, feathers, scales. They're all made out of different versions of keratin, and some other proteins that provide strength and resist tearing in cells. And those, the intermediate filaments, because keratin is uniquely found in animal cells, appears to be only part of the cytoskeleton of animal cells. We don't think it's found elsewhere. So here is a photo of cells where the microtubules have been stained with a dye that colors them either a yellow color or white color. And you can see that it is a highly complex network of these fibers that are made up of two proteins called alpha and beta tubulin and form hollow tubes. Where we're going to see these particularly playing an important role is when we talk about cell division, because they give rise to a structure called the spindle that guides the movement of the chromosomes during the initial part of cell division. But see, that's what microtubules do. Not only do they provide a structural framework, but they also supply a framework for the movement of things. So that's the role of microtubules. So when we see something like an amoeba, and you can find any number of YouTube videos of amoebas, and I believe I've actually posted an amoeba video in the resource page, so you might want to check that one out too. Amoebas are cool to watch under the microscope because they're constantly changing shape. But that constant change in shape as they explore their environment, well, that's driven by the microtubules of the cytoskeleton. So here you see microtubules doing what microtubules do best, or one of the things that microtubules do best. That is not only maintaining the shape of the cell, but changing the shape of the cell along the way as well. Their other role, framework for movement. So a roadwork or a roadway or a trackway for things to move on. Cilia and flagella are extensions of the microtubule part of the cytoskeleton, because when we look inside the cilia and flagella, and the only difference between the two is the length. Cilia are short and wave backwards and forwards. Flagella are long, but when the base of the flagellum waves backwards and forwards, it propagates what's called a sine wave or an S wave down the flagellum, something that you couldn't do on a cilia because it's too short to do that. That's the big difference between cilia and flagella. In terms of cross-section, and this is an animation that is available to you through the resource page for Chapter 4, that shows not only the classic 9+2 structure, so 9 pairs of microtubules in the rim and 2 microtubules forming the core, linked together by spoke proteins, and then the microtubules in the rim are connected by motor proteins called dynein. And the dynein pulls against the adjacent microtubules, but because the ends of the microtubules are anchored, this doesn't move, and so that causes the cilia and the flagella to go like this because of the alternate pulling, and that's illustrated in this animation. So as I said, this animation is available to you in the resource page, so I'd strongly suggest you go and take a look at it there. And you can see how the cilia and the flagella move backwards and forwards like this. If you have errors in cilia function, flagella function, then in the case of some men, it's a cause of their sterility, because if the sperm can't swim well or are paralyzed in some fashion, they can't move from the vagina up through the uterus, up into the fallopian tubes where fertilization actually occurs. So having non-functional flagella could be a reason for infertility. Another problem with cilia is that cilia line the bronchial passages that lead to the lungs, and the smoke from cigarettes and tobacco in general paralyze those cilia, which is the reason why every morning a smoker has to cough furiously to get all the accumulated phlegm out of their lungs, because normally the cilia in the bronchial passages would simply sweep that material up into the throat and you would swallow it overnight. It wouldn't accumulate. This is the material that you're constantly swallowing during the day. You can feel it, the viscous material that comes up through your throat, and you have to swallow it occasionally. That's the material that's being swept up out of your lungs. It keeps the lungs clean and allows for efficient gas exchange. But in smokers, those cilia become paralyzed. The good news is that if you quit smoking, those cilia quickly reestablish themselves and start functioning normally again. The other interesting place where inactive cilia become a cause of a condition is in a condition called situs inversus. This is where the orientation of the organs in our body cavity, the heart, the lungs, the liver, the digestive tract, are rotated through 180 degrees. So instead of the heart being offset to the left, it's offset to the right. The liver is offset to the right instead of to the left. The way the digestive tract is folded up inside the lower body cavity is reversed around. The reason why this happens is because the cilia that line our body cavity set up a clockwise fluid flow in the body cavity during our early development. If those cilia don't set up this fluid stream, then situs inversus takes place, because the normal location of the body organs is a function of that flow of fluid around the body cavity. If it doesn't exist, then the organs are arranged in that 180 degree reversed set when we talk about situs inversus. This is typically diagnosed when fairly early on, because a physician will be listening for the heartbeat offset to the left, can't find it, moves the stethoscope over to the right and picks up the heartbeat, and immediately the diagnosis is situs inversus. It is a rare condition. If you happen to have this, and you probably know if you have it, it's not a big deal. It's not going to affect your ability to live a normal life. You can live a normal life. The only time that it may become an issue is if you have appendicitis, for instance, because the pain is not going to be offset to the right of your body cavity, it's going to be offset to your left. So it may get misdiagnosed initially, hopefully not, particularly if you know that you have situs inversus. You can let the physician know that you have it, and then they know what to look for. Or if you are unfortunate enough to require a transplant, it makes plumbing in, particularly for a heart transplant, the new organ, a little bit technically more awkward, but it can be dealt with. So you shouldn't panic if you've been diagnosed with situs inversus. You're going to live a normal life, and there's not going to be any domino effect because your organs are rotated through 180 degrees. But we know what causes it. It's because of the paralyzed cilia that line the body cavity. That is not mentioned in the book, but that's something that I wanted to bring up for you because, again, it's an example of the cilia playing an important role. So, last concept check, and it's an easy one. Compare and contrast cilia and flagella. 30 seconds of jeopardy. So you may have noticed that my 30 seconds is probably not really 30 seconds, probably a lot shorter than that, but that's okay. So how do we compare and contrast cilia and flagella? Well, the contrast, of course, is the length. Cilia are short, flagella are long. And so in terms of the way they move, the base waves backwards and forwards, but that's all the cilium does. But in a flagellum, the waving of the base sets that S wave or sine wave running down the flagellum that allows for the movement of the cell. You also need to remember that they have the same cross-sectional structure, the so-called 9+2 structure of microtubules connected by dynein motor proteins in the rim and connected by spoke proteins to the central pair of microtubules, which is the +2 part of it. The other thing that is not clearly mentioned in the book, but which I want to make you aware of here, is that these really are an extension of the cytoskeleton. They are an extension of the cell itself, because if you look really carefully at a good electron microscope image of a cilium or a flagellum, you can actually see the cell membrane encapsulating the microtubule complex that is the core of the cilium or the flagellum. It is fully enclosed within the plasma membrane or the cell membrane of the cell. It really is an extension of the cytoskeleton, an extension of the cell itself. So that's something also to remember about cilia and flagella. It's not something that is pointed out particularly well in the textbook, but I think it's something that I want you to be aware of. Okay, so let's come back and revisit bacteria for a minute. We know that they are really problematic because a lot of human diseases are caused by bacteria. And those that are not caused by bacteria, the vast majority are caused by viruses. So between them, bacteria and viruses account for a large number of human diseases. And then there are fungal diseases. But in the case of bacteria, we've always felt that the way we deal with them is through antibiotics. The problem, of course, is that they adapt to those antibiotics and become resistant. They evolve. Well, it turns out that humans can also evolve to become resistant to bacteria. So long as the exposure is an extended exposure, people can actually become resistant to the bacteria. So the example that is cited in the textbook is the bacterium that is responsible for cholera in Bangladesh, what used to be called East Pakistan. It's on the east side of India. What is now Pakistan proper used to be called West Pakistan. They used to be a single country when the Indian subcontinent was first divided up by the British when they gave the area independence. So they created two blocks, West Pakistan and East Pakistan, for individuals who were Muslim, and India proper was supposed to be a Hindu country. It turned out that the people of East Pakistan and West Pakistan did not see eye to eye. They are very different peoples. They just happen to share Islam as their adopted religion or their major religion. And so there was war fought, and eventually East Pakistan and West Pakistan went their separate way. West Pakistan became Pakistan, and East Pakistan renamed as Bangladesh. The Indus River flows across the top of India and empties into the Indian Ocean through a large delta in Bangladesh. And it is in the Indus River Valley that the cholera bacterium has lived amongst humans for a long period of time. We estimate somewhere between 20 and 30,000 years. Now, cholera is a lethal bacterium. It produces a toxin that causes the cells that line your digestive tract to leak fluids. And that changes the contents of your gut. It becomes more fluid and you get diarrhea, but it also means that you become severely dehydrated, and cholera kills you through the dehydration. And unless you treat the bacteria, you're not going to treat the diarrhea. You can't treat the diarrhea without treating the bacteria. It turns out that amongst people who live in the Indus River Valley, there is a mutation that involves one of the proteins that's responsible for the fluid flow. And there is a relatively high incidence of this mutation amongst people who live in Bangladesh. Because over the period of time that the cholera bacterium has been an issue for humans, since it started infecting humans, a number of mutations have occurred, and the ones that were beneficial to humans in terms of resisting the effect of the cholera bacterium became more and more frequent in the population. And because the humans and the cholera bacteria have been living together or coexisting together for a long period of time, it means that more and more people have inherited this mutation over time, so that a significant number of people in the Indus River Valley are actually resistant to the bacteria. So if individuals can spontaneously develop these types of mutations, it is possible for humans to become resistant to bacteria. We believe that the mutation that is responsible for cystic fibrosis actually started out in the Indus River Valley, because it affects a protein that regulates chlorine movement across a membrane, which also involves fluid movement. And the presence of this mutation actually provides some resistance to gut bacteria, particularly the cholera bacterium. But of course, if you happen to have cystic fibrosis, that's bad news, because it will eventually wind up killing you, because it is a lethal condition with no cure. But the reason why the mutation exists is because, again, we have 2 copies of genes for all of these characteristics that we possess. And if you have 1 copy of the normal gene and 1 copy of the cystic fibrosis gene, you actually have a resistance to the cholera bacteria that you otherwise might not have if you just had the 2 normal genes. Of course, if you have 2 copies of the cystic fibrosis gene, then you have cystic fibrosis. You'll learn more about this when we do the inheritance portion and the genetics portion later in the semester. The same sort of argument has now been documented, or the same idea has been documented for a group of unique individuals who have a resistance to the HIV virus. The virus normally enters cells that are part of the immune system, called the CD4 cells. We've talked a little bit about this before in the previous presentation. And so this is just another example of where people become resistant to bacteria, because remember, the mutation that provides the resistance to the HIV virus originated amongst individuals who were resistant to Yersinia pestis, the plague or Black Death bacterium, back in the Middle Ages and the Dark Ages, when the plague bacterium first arrived from Asia in Europe. Originally, it was very, very lethal, but over time, over about a period of 300 years, individuals emerged that had resistance genes, and it would become the same resistance pathway for becoming resistant to HIV virus. It would be the same mutation that provided resistance to the Yersinia pestis bacterium that would provide resistance to the HIV virus. So humans can adapt to bacteria. Just as bacteria can adapt to our antibiotics, humans can evolve resistance to bacteria so long as there is time and so long as the mutations offset the effect of the bacterium. So long as the bacterium remains virulent and can be deadly, and there is no mutation that provides a resistance to the bacterium, then the bacterium remains a virulent, deadly bacterium. But if mutations appear in humans that provide an advantage, that is, provide a resistance to the effect of the bacterium, then that mutation is going to become part of a larger, or become a larger part of the population's genetic makeup, as long as time continues to be available for that to be passed on from one generation to the next. As long as it's inherited, that is the possibility. So that's our discussion of the general structure of cells, focusing particularly on eukaryotic cells, but of course also showing the structure of a typical prokaryotic cell. When we come back to the next chapter, we're going to focus more in on membranes and their roles in cells, and focusing not only again on the structure of the membrane, but also focusing on movement of materials across cell membranes. Because we do know that cells import and export materials, molecules, ions, into and out of cells. And it's important for us in terms of understanding cellular structure to understand how that takes place. So that will be the topic of the next presentation. So until then, if you have any questions, concerns, or comments about anything that's been covered in this chapter, or if you want to get some help clarifying things from the 3 previous chapters that are on the first exam, by all means, come and see me in office hours. I'll be happy to help you out. Otherwise, I'll see you back here again when we talk about membranes and the function of membranes. Bye till then.